Hi, I'm Fiona from the National Centre of Biotechnology Education based at the University of Reading. Thank you for joining me for our second video in our protein module. So in our last video, we discussed how DNA is transcribed into RNA and how RNA is then translated into proteins. So what happens next? Well, once the RNA has been translated into a chain of amino acids, it needs to fold into its functional protein structure. So the polypeptide chain is what is known as the protein's primary structure. Two amino acids joined by a peptide bond are known as a dipeptide. Multiple amino acids joined by peptide bonds, which are obviously formed by condensation reactions, are known as a polypeptide. So the polypeptide chain, once it has been translated, can then fold into its secondary structure. Some can do this unaided and others need chaperones to be present. Features of a protein secondary structure include things like beta sheets and alpha helices. Both the beta sheets and the alpha helices are formed by hydrogen bonds between amino acids within the polypeptide chain. So the oxygen atoms from one carboxyl group of one amino acid can form a hydrogen bond with the hydrogen atom on the amine group of another amino acid. So you can see this here where there are two polyglycine chains. So these are chains of the amino acid glycine, and they have lined up with each other, and hydrogen bonds have formed in between, creating a beta sheet secondary structure. So depending on the side chains and how these align will depend on whether there are areas of beta sheet, where there are polypeptide chains that lie down next to each other in a sheet-like arrangement, and the side chains will be above and below this arrangement, or whether they form a helical arrangement with hydrogen bonds between one amino acid and another far down the chain, and this arrangement causes the side chains to stick out so they are free to interact. So these then fold further into the protein's tertiary structure. So a whole host of non-covalent bonds are involved with this, and one type of covalent bond, which is the disulfide bridge, which is much stronger than the other bonds present in the tertiary structure, and it helps to stabilise the protein. So cysteines must be present there in order for this to form. So for example, the prion protein is shown here, which as you can see has some areas of alpha helices and beta sheets, and the beta sheets are shown in turquoise. And importantly then, in pink, you can see there is a disulfide bridge, and this is formed between two cysteine molecules. So this bond is very important to the prion protein structure. Without it, it behaves very differently. So the tertiary structure then is mainly down to the interactions of amino acid side chains that are present for that protein. So whether the side chains are similarly charged and repel each other, or oppositely charged and therefore attract. So non-polar hydrophobic side chains tend to group together inside the protein structure, leaving the hydrophilic side chains on the outside to interact with the mortar molecules. Some proteins then only have a tertiary structure, and they are functional like that. Others, however, have multiple subunits that come together to form its quaternary structure. So for an example, hemoglobin is one such protein. So this has 4 subunits. So it has 2 alpha chains and 2 beta chains. Now these come together to form the quaternary structure and the functional protein hemoglobin. Each subunit of hemoglobin contains a heme molecule that contains iron. Now this allows each subunit to carry an oxygen molecule. So hemoglobin can therefore carry 4 oxygen molecules in total. Now this structure allows it to maximise how much oxygen it can carry around the body. So hemoglobin is a globular water-soluble protein, which is obviously very important and allows it to be able to travel around in the blood, delivering oxygen to where it is needed. So this is an excellent example of how a protein structure is directly related to its function. So just to explore a bit further how the structure and function of a protein are related, we will now have a little look at collagen. So collagen is the most abundant protein in animals. There are lots of different forms that have different properties. So collagen provides our skin with elasticity, our tendons and ligaments with structural support and strength, and it also supports our connective tissues. So the structure of collagen is very different from hemoglobin. So for a start, it is fibrous instead of globular, and it is also insoluble instead of soluble as hemoglobin is. So it is formed of very long chains of amino acids with repeating sequences. So this allows it to form long helical structures as its secondary structure, and these are known as alpha chains. So these alpha chains form a triple helix, and tertiary structure then go on to form even larger helical structures that are even stronger. So these then have covalent bonds between them, and this forms very long, very rigid chains. And these are obviously perfectly suited for the strength and stability needed throughout the body, which obviously collagen provides. So this is obviously another excellent example then of how the structure of a protein is directly related to its function. So what happens when things go wrong? Imagine if there was a mistake in the DNA. So this could be a deletion, an insertion, or a substitution. A deletion is when one base or more than one base is deleted. An insertion is when one or more bases are incorrectly inserted into the DNA sequence. And a substitution is when one or more bases are substituted for another. So maybe there should be a T, and instead this is swapped to a G, for an example. So these sorts of mistakes can occur when the DNA is replicating. So obviously if this occurs in an adult cell, then that should only really be confined to that cell. However, if a mistake is made during development of a fetus, for an example, or an embryo, then this could be present in all the cells or many of the cells, depending at what point in development it occurs. Now, if the mistake results in an essential protein being non-functional, then the development of the embryo will likely not continue. If it results in a protein that can function, but less well than the wild type, then development may continue, but the resulting baby will likely have a lifelong condition, and that will obviously vary in severity depending on what protein was affected and how seriously it was affected. So let us look then at how a mistake in DNA goes on to affect the protein. If a deletion occurred in the DNA, here we are looking at just one base being deleted, then when the messenger RNA is transcribed, there would obviously be a base missing in the RNA. So you might think that this wouldn't cause too many problems, it's only one base. However, when we look at how the RNA is translated, with the code being translated obviously in triplet sequences, then even one base missing can have catastrophic repercussions for the protein. So let's have a look then at what actually is happening. So if this thymine was to be deleted in the DNA sequence, then this adenine would obviously not be transcribed as it should be in the RNA sequence. So this causes what is known as a frame shift. So this means that all the bases from this point onwards shift over by one place, meaning that the codons are now all different from this point onwards. So the first two codons are still the same, but now since all the bases have shifted along by one, instead of this codon being translated into asparagine, it is now going to be translated into threonine. And this is not the only change, since as we look further down the chain, we can see that this has caused many changes to the amino acid sequence. So this is obviously now not going to be translated into the intended protein. Now, frame shift mutations tend to be the most severe, causing the production of an amino acid chain that, if it bears no function, will likely be degraded, or sometimes it may even have a harmful function or obstruct the ability of other proteins to do their function. Sometimes mutations are not quite so extreme. A whole codon will be deleted, which instead of causing a frame shift, instead only that amino acid will be missing from the chain. This can still result in a functioning protein, albeit perhaps slightly truncated. Substitution events can cause a minor change to the protein or can have a major effect. So, for example, when a base is substituted for another, it can result in a single amino acid chain. So, depending on the properties of that amino acid will obviously then affect how different the protein is. Or sometimes one base substituted for another can result in a new stop codon. So as you can see here on the DNA strand, if an A at this point would mutate to a T, then this would mean that the base on the RNA would be an A instead of a U. So this would result in the codon being read as UAA instead of UAU. And UAA is a stop codon. So the amino acid chain would stop here, obviously resulting in a very truncated protein, which would likely have no or very little function. So you can start to see then how important it is for DNA to be replicated correctly and for the RNA therefore to be transcribed correctly. Of course, sometimes a mutation can occur that actually confers an advantage, and these chance mutations are of course critical for evolution. However, let's go back to when things go wrong, and let's take an example of hemoglobin and sickle cell anemia. So as I mentioned earlier, hemoglobin is made up of 4 subunits, so 2 beta polypeptide chains and 2 alpha polypeptide chains. Now, actually, with sickle cell anemia, all it takes is one substitution at one base, and that is enough to cause this disease. So in the wild type hemoglobin, the DNA, one of the codons should be CTC. But if it is changed to CAC, which is the case in someone with sickle cell anemia, then this, when it is transcribed into the RNA, is instead of being GAG in the wild type, it then becomes GUG with sickle cell anemia. So this is then, when translated into the amino acids, means that instead of glutamic acid at this point in the amino acid chain, which is what it should be in the wild type, it actually is valine. Now, valine and glutamic acid have quite different properties, and this therefore means that instead of the hemoglobin being able to form its globular structures, it actually now forms long rigid rods, and this obviously therefore has very, very different properties. And in sickle cell anemia, this causes sort of sickle-shaped cells to form, which are characteristic of this disease. So these sickle-shaped red blood cells that are formed because of this mutation, therefore have very different properties to what normal hemoglobin has. And they don't very efficiently carry oxygen, and they can actually, because of their shape, block capillaries, which then means that oxygen is cut off from various tissues. So this causes then serious pain and serious health problems for those with this condition. Interestingly, however, this mutation persists because actually those that are heterozygous for this mutation, so they have one copy of the mutation and one copy of the wild type, actually have resistance to malaria. So in areas of the world where malaria is still rife, those that are heterozygous for this mutation actually have quite an advantage, and therefore obviously go on to pass on the mutation to their offspring. Okay, so today we have been through protein structure and function, what happens when mistakes are made in the DNA sequence. I hope you now understand how important it is for DNA to be replicated correctly. So next time then, we are going to be undertaking our NCBE at-home protein experiment. So normally, if you were studying proteins in the classroom, you might look at proteins and run what we would call a protein gel, which the NCBE provide resources to do. But since we're all at home, we're going to instead have a little look at how proteases work, looking at some fresh pineapple and some jelly. So you just need to try and get hold of some fresh pineapple, some jelly, and if you have some tinned pineapple too, then that's also a nice addition to the experiment. So I really hope you can join me then, and I hope you've found 2 protein videos useful. So see you next time.